you. Let me share my screen with you. All right. So hopefully now you can all hear me and see my screen. Yeah? Can I get like one thumbs up, Kim, just to make sure?
Yep.
Yeah, okay, great, both Kims, awesome. Okay, thank you very much for the introduction. I'm happy to be here with you today virtually to share with you a topic that is very near and dear to my heart, and that is because my lab at GVSU researches the hygiene hypothesis in immunity. So what I'm going to do is I'm going to walk you through what the hygiene hypothesis actually means, and at least in my opinion, and what that means for immunity, what we know kind of so far. And then I'm actually going to walk you through experiments that we've actually done in my lab and show you unpublished data that's been generated by both me and my students, and kind of tell you where we're at in our own research at GVSU. So you're seeing kind of really fresh, science in action in the second half of this talk. All right, so let's just get started. And this illustration that I have on the beginning slide here, you'll see that again, and I'll explain a little bit more about what this means later. All right, so I just want to maybe refresh you about the goals of the immune system. So if you attended my previous talk in the summer, this slide is kind of going to be a refresher for you. But I want to remind you about what the goals of the immune system actually are. So the first goal of the immune system is to recognize danger if it enters your body. So to realize that there is potentially a threat and maybe we have to do something about it. Once that danger is recognized, your immune system wants to contain the threat as well as potentially eliminate that threat. We need to get rid of it, right? We need to recognize that there's danger and we want to get rid of it because the danger that the immune system needs to deal with potentially could cause a lot of harm or even death to the host. So the immune system's goal is to get rid of the danger. And then a point that some people kind of forget, including some of my fellow immunologists, it's actually very important for the immune system then to turn off when the danger has been cleared away. If you don't have an immune system that turns off properly, you actually can have some problems that can occur. And then finally, the goal of the immune system is to make what is called immune memory. And this concept, this immune memory right now is super hot in the field, just in general, because that is a goal of a vaccination. A vaccine is going to try to make immune memory in order to keep you safe later on if you happen to see this danger, whatever it is, again. So what is the immune system? Well, the immune system is made up of your white blood cells. So in the blood, there's two main types of cells, red blood cells, and in this picture, those look like the little red discs. Red blood cells are what carry oxygen. That's their goal, very important. The white blood cells make up the other part of the cellular component of the blood, and that is immune cells. And sometimes we just say white blood cells and we just kind of lump everything together, but actually it's quite complicated. There are many different types of white blood cells that make up your immune system. So on this next slide, I actually took a picture from one of the immunology textbooks I use because it shows some, not all, but some of the different types of white blood cells. So all of these cells that you can see are actually all white blood cells. You would call them all white blood cells, but they're actually different types of cells. And so if you look over on this side of the slide, we have three cell types called eosinophils, basophils, and neutrophils. These cells are called granular leukocytes because they're full of these little specks. Can you see those little specks in these different cell types. Those specks are called granules, and they help these cells carry out their immune function. We're not going to talk about all of these cells in detail. I'll kind of point out the ones that are most important as we go along in the talk. So we have these granular immune cells here. And then we also have white blood cells that are not granular, meaning that they don't have those little pinpricks that you can see by microscope. So up at the top here, we have lymphocytes, and lymphocytes are things like T cells, as well as B cells. T cells and B cells are extremely important for the immune system. We're not going to talk a lot about B cells in this particular talk, but we are going to talk more about T cells. So we'll get more detail in a little bit about that. One thing I will say about B cells, however, is that B cells are what produce antibodies. And B cells that produce antibodies make these other cells that are called plasma cells. If you've ever heard of that, I don't know if you've come across the word plasma cell before, but plasma cells are basically antibody producing factories. So if you've got a lot of plasma cells around, your body is making a lot of antibodies. Then we have monocytes. And I'm just pointing out monocytes because it's important to note that monocytes are in the blood. And when they exit the blood and go into the tissue, like the skin or one of your organs, then we call them macrophages, because as immunologists, we just like to be super confusing with the names. I'm just kidding. Things are discovered at the same time and different names are given to them, and then we just have to call them two different things. So the two cells that we are going to talk more about in terms of my lab's research are going to be these T cells as well as macrophages. So we'll get there, and when we get there, I'll give you some more detail about those cells. All right, so still kind of on an immune system refresher right now, okay? So we know the goals of the immune system. We know that there are lots of different cell types that are in the white blood cells. But what actually happens in an immune response? I like to think of the immune system as a potent weapon that specializes in host defense. So what does it have to defend against? It has to defend against those things that I was just calling danger in the first slide. But what that danger actually is are infectious agents, which we in microbiology and immunology give a special term to. We call those pathogens. So pathogens are organisms that are capable of causing disease in humans. And so in this picture, what I have showing here are two of the most common types of pathogens. Up at the top, we have a virus. So this would be something like SARS-CoV-2 that causes COVID. That's a virus. And then down here, we have a bacteria. There are many bacteria that cause diseases. For example, E. coli is a bacteria you may have heard of that can cause gastrointestinal distress. There are many types of viruses and many types of bacteria that can cause disease in humans, and we would call those things pathogens. So what happens during immune responses? I like to think of things in graphical form because as a scientist, that's just how my brain works. So sorry, this is what you have to deal with me. So what we have here is a graph that I just made where we have a number here on the y-axis. So this is going to be like increasing numbers of something up here closer to the arrowhead and decreasing numbers of something on the bottom as we get lower. And then we have time. So over time, what's going to happen? So let's pretend that this is in this poor guy's body and he gets one of these pathogens. Let's say it's the virus. So in this human's body, the virus, in this case, in our example, is going to replicate. So the number of virus particles in the body is going to replicate. This is what the virus does. This is how it causes sickness, okay? So as the virus replicates, the number of these virus particles is going to increase, which is why you see this red line representing the virus going up on the scale over time. Lucky for us, the immune system comes in and can recognize the danger. When the immune system recognizes that there is a danger happening, the immune system also starts to replicate and increase to kind of meet the threat. One of the white blood cells that are involved really early on in the response is the macrophage. If you've heard of the terms innate and adaptive immunity, innate immunity means the immune system, part of the immune system that responds first. So the macrophage, for instance, would be part of this innate first responders. Macrophages, they are usually able to respond to an infection within an hour, which is pretty cool. They are lining kind of all of the areas of the body that are potentially open. So underneath your skin, you have macrophages. And that's why they can respond so quickly. We'll talk more about what they do later, but for now, just know that macrophages are responding very quickly to the pathogen. So what happens? Well, unfortunately, in our case, the macrophage isn't good enough. So the pathogen keeps replicating. So it's continuing to increase, even though we have the immune system responding to the virus. Lucky for us, the immune system's not done. The immune system can call in the big guns at this point, which are things like T cells, which we'll talk more about later, and B cells. And remember, B cells are producing antibodies. So T cells and B cells can respond next. And these T cells and B cells are very, very good at what they do. So what happens, hopefully, in the immune response is that with the T cells and B cells now responding, we can now contain the virus and actually start killing it off, so that we actually will eventually completely remove the virus from our bodies. That's the goal of the immune system. So what happens to our immune cells that have, replicated and basically created an army to get rid of this virus? Well, we don't want to keep them around if they're not needed anymore. So this is called contraction, actually, in immune lingo. And during contraction, most of that immune response will die, but not all of it. So you'll keep some of them around, potentially for a really long time. And we call that the memory. That's immune memory. So let's say it's a few years later and this guy, unfortunately, gets reinfected with that same virus. The virus starts to replicate again, but this time, the immune response is so fast and so massive that it can very quickly kill off the virus and get rid of it. This is the goal of memory, because the memory response acts so quickly in your body that generally you have no idea that you even should be sick. It gets rid of the virus before it can cause a problem. So in terms of vaccination, including COVID vaccines, what we're trying to do is make this memory so that if you get infected with the actual virus, your memory can respond fast enough that you do not get sick. Okay? And then we have the same type of cycle where a lot of the immune system will die off again, and then you'll still have memory. So this is how an immune system is supposed to respond to something like a virus or a bacteria infection. This is the ideal, right? And the immune system is very, very good at doing this. You know what, this might be a time before we move on to the next phase. I don't know if there's any questions at this point. You can also, wait for the end if you prefer to do that. That's totally fine. We're going to kind of start changing gears a little bit here, so I want to make sure we clarify anything.
Kristen, I have a question, just very, very generally. How does the system recognize a pathogen that's been in the system before?
So the way it does that is through receptors that are on the cell surface. So I didn't draw them, but you can picture all along the outer edges of this B cell and this T cell, they have specialized receptors that stick out. And those receptors can recognize and bind to, so it actually like interacts with bits and pieces of the pathogen.
Okay, great.
So it's very specific.
One follow-up to that, then when there's a reinfection of a prior pathogen, it's primarily the B cells or the T cells that do the work?
Yes.
Okay.
Only B cells and T cells make memory. They're
Okay.
very specialized. So yeah, yep.
We have a question on chat from Greg. Does the second pathogen exposure increase the immune memory level?
Yeah, it can. So we call those things, like primary memory, secondary memory, even tertiary memory, because obviously in some places, you'll have like cyclical infections. I'm thinking of like malaria, for instance, where potentially you can be reinfected many times with the same thing. And we see that there's like, we call it boosting of the memory. So that's actually why you might also get more than one shot of a vaccine, is because after the first booster, you might get this, level of memory. If you give another vaccine dose, then this memory level after that vaccine might be up here, if that makes sense. So yes, that can happen. Good question. Any others? Are we ready?
Looks like we're ready.
Okay, great. So coming back to this idea, right? The immune response is a potent weapon, especially against dangers that come into our body, like pathogens. ... However, the immune response is also a potent weapon, even when things go wrong. So this is the problem. If the immune system is not working properly, it can still be just as potent of a weapon, except this time, it might not be doing damage to a pathogen, it might be doing damage to the host. And so an example of that would be allergies. Allergies are when the immune response is using its potent mechanisms, but unfortunately, in the wrong situation. And that causes allergies. So that's where we're going next. We kind of know a little bit about how the immune system works when it's supposed to be working, right? But now let's talk about what happens with the immune response in allergies. And if any of you suffer from allergies or know people who suffer from allergies, you know, I'm going to tell you actually what is happening in your bodies at the cellular level to make you miserable. Okay. All right. I got to introduce a new cell for you. We haven't talked about this one yet. A mast cell. I don't know if anyone's heard of a mast cell before. If you have really significant allergies, you may know what a mast cell is because you've had to Google it. Mast cells are actually kind of like macrophages. They're in the tissues and they line the areas of the body that potentially have contact with the outside world. So once again, they are in the epithelial layers, which basically just means they are right underneath your skin. They line that. Right now, you have mast cells lining underneath your skin. And then they're also in the mucosal tissue. And so what I mean by that is any part of your body that is basically like a tube where either air or food or liquid passes through. That usually has a mucosa, like mucus surface, that lines it. So you have mucosal surface in your nose. You have mucosal surface in your throat. You have mucosal surface in your lungs. You have mucosal surface in your intestines. So you've got a lot of mucosal tissue. So mast cells live in that mucosal tissue too. What do they normally do? Well, they did not, you know, arise to cause allergies. That is not why they're around. They do have normal functions in people, and people who don't have allergies still have mast cells doing something. They function during normal immune responses to pathogens. And they also have this really cool job of repairing damage. So like if you get a cut or something on your skin and you need to, that cut eventually repairs, right, through wound healing, mast cells can be involved with helping to do that wound healing. So they actually are pretty important. Here's a picture. I love this picture. Here's a picture of a really, really close close-up of a mast cell, okay? So this kind of round thing right here, this bigger round thing, that is one mast cell. They're big cells. And mast cells are granular. Remember how we talked a little bit about granulocytes? So you can see like all of these dark areas. I'm outlining a few with my mouse here, which hopefully you can see. All of these like dark areas are chock full of lots of potentially toxic chemicals and compounds. And the mast cell just holds on to them in case it needs them. Mast cells are really distinctive under the microscope. Okay. So mast cells, what are they actually doing in allergies? Allergies have two stages. So you may have heard before that, I don't know if anyone's heard like the saying that the second time you're exposed to the allergies is actually the worst. Has anyone heard that before? Maybe? I don't know if you've heard that, but some people may have heard something like that. Like if you are allergic to a bee sting, let's say, if you get stung for the very first time by a bee, you're going to be okay. But it's actually the second time that you get stung, maybe. And that's actually true. What happens in the immune system. So this first stage of an allergic response is called sensitization. And so what happens is we have an allergen. An allergen is something that can cause allergies. So maybe this is pollen or dust or something like that. And in my picture, the allergen looks like a little moon or something like that, little gray ball. So we have an allergen, and this allergen will actually involve a B cell So B cells can be activated by allergens. And then B cells that are activated by allergens make a special type of antibody. This is called IgE. It's a special type of antibody. Sometimes people who have allergies, sometimes you'll get IgE measurements in your blood to see if you have IgE in your blood. That if you have increased amounts than normal of IgE, that can be signs of an allergic response happening. The thing that's really special about IgE is that it actually can bind to a mast cell. But it's really weird how this antibody binds to a mast cell. These antibodies bind to this mast cell kind of facing outwards. And so I only drew one here for simplicity, but you should imagine that this mast cell is covered with this antibody all around on the cell surface. And at that point, if you never got exposed to the allergy again, you should be fine. Those mast cells aren't going to do anything. They're holding on to their granules in the inside. Nothing's going to happen. But what happens if you get exposed again? This stage of an allergic response is called degranulation. So what happens is you get exposed to the allergen again. That allergen can bind to the antibody that's on this mast cell. This causes the mast cell to rapidly release its granules. That's called degranulation. So this mast cell is basically just spewing out what's in its granules. Guess what's one of the main components of the granules? Histamine. If you've heard of histamine or have had to take like Benadryl, which blocks histamine from working, one of the main components in mast cells that they're going to spew out at this point is histamine. There's also a whole bunch of other things that get released from a mast cell, which causes all the things that make you miserable during allergies. So these are things that will cause inflammation. It causes vasodilation, so the blood vessels kind of dilate, which can cause more fluids to be exchanged, which actually can cause the itchiness and the sneezing. It causes muscle contraction. Like basically, if you've ever had allergies, picture how you feel when you have allergies. It's due to these granules being released and causing havoc. The problem, though, is you can maybe imagine that, let's say you get exposed to allergen again. You're just going to make more mast cells that are ready to degranulate and more degranulation. More and more and more and more and more. This is why allergies can get worse over time. Now, I'm going to sneak in two other cell types real fast here at the very bottom. Eosinophils and basophils. These are very rare white blood cells, but they are also involved in allergies. I know some people, if they're getting blood work done to check for allergies, they'll look for instance, the amount of eosinophils in the blood. And if those are increased, then that also can show that there's potentially, an allergic response happening. Eosinophils and basophils are very similar to mast cells. They have granules, they promote an allergic response. So this is what's actually happening at a cellular level when you have hay fever, for instance. The mast cells in your nose are getting triggered and degranulating and causing your symptoms. Okay. So you can have localized allergies that go away, but unfortunately, you can also have something that's called chronic allergies, meaning it does not go away. And I think probably the most well-studied chronic allergy is asthma. So asthma happens when you have what I just described on the previous slide happening in the lungs. And what happens, if you look here in this picture, we've got like on the left, we've got the normal looking lung, and on the right, we have an asthmatic lung. And what happens in asthma is that you get chronic inflammation. So you get this mast cells and basophils and eosinophils just continuing to respond, continuing to cause inflammation. And then what you get is muscle tightening due to what those cells release out of their granules. You get swelling, and you get excess mucus. And if there is an exposure to whatever the allergen is, you can get an asthmatic attack, because if that allergen, is enough, if it gets down there, it can cause just kind of a brief period of degranulation that's just massive. And that's what an asthmatic attack is. So unfortunately, this is chronic. It does not go away. We don't really have any great treatments for asthma other than just to control the symptoms. So that's why there's actually quite a few people who research asthma to try to figure out ways that we can either prevent or treat this better. All right, let's see where we're at. I think I'm going to keep going, but we'll probably stop in a few minutes to see if there's any need for clarification or anything at that point. So this got some scientists to thinking, what causes allergies in some people, but not other people? Why are some people getting allergies and some don't? So for example, in my family, my husband and my son both have, classic type hay fever type allergies. They're seasonal. They're worse in the spring and the summer. My daughter and I do not have those allergies. So, why we live in the same house, like why potentially are some people getting these allergic responses and some don't? And so there is one avenue of thought about genetics. So your genes potentially make you more susceptible to allergies. And that's actually true. We know based off of a bunch of research that there are certain genes in your DNA that are linked to higher risk of asthma and allergies. That's called, if you've ever heard the word atopy, atopy means, an increased risk of allergic responses. So we do know that. But what I'm more interested in is, how about the environment? So this is kind of a nature versus nurture, right? So nature, as in the genetics, what are you born with? But what about nurture? What about your environment that you grow up in? Some of the things you can't control, obviously, but what about those things? And we know a lot less about how the environment that you grow up in impacts your allergic risk. So we turn to some really actually kind of old literature. And we found that there are a lot of environmental factors that influence your immune response. Now, these different factors are very important, especially during childhood. 'Right after birth, until about 8 to 10 years old or so, is when the immune system is still developing and is still very heavily influenced by the environment. So what are we talking about here? What impacts the immune system of a young child? What the child eats. Are they consuming breast milk, formula, one or the other? One of these things is not necessarily better than the other. It's just that they can influence the immune system developing. Your family size. How many siblings do you have? For instance, do you go to daycare or not? All of that can influence the immune system. And then we have over here your infection history. So does this child get infected with pathogens? Does this child receive vaccinations? And what does the environment look like in terms of animal exposure? Does a child live on a farm? Or does a child live in the city? Do they have pets? These two things in particular are very interesting to me, because when I look at this, I can see that microbes like bacteria and fungus and viruses, in both of these situations, these microbes could be influencing this child's immune system. And what's really interesting to me is that what happens early on in this kid's life might have lasting consequences for the immune system. In my lab, we call this microbial experience, which is a very broad term that we made up to encompass these two areas that are highlighted by red boxes. So what types of microbes, like bacteria, viruses, fungus, for instance, is this child getting exposed to early on, if any? And what does that mean for the immune response? So microbial experience, that's a word we use all the time in my lab. So these differences here from one child to the next, to the next, to the next, are obviously dependent on many factors. I mean, location, economic status of the country or nation, as well as, the family, healthcare availability. I mean, there's a lot of things that feed into what this microbial experience is for a kid. So let's look at this map. So in this map, we have in red showing the countries that have most parasitic diseases. So parasites, these are things like worms and other types of parasites, where these types of infections are more prevalent. And you can see that these are mostly developing nations. That's where we tend to see more of these types of infections. They're usually less industrialized, less access to healthcare for the majority of the population, etc. What was really interesting is if you look at these same countries in red that have those increased parasitic infections, they have very, very low allergies and asthma. In fact, there are some countries that are pictured here that it is extremely rare to get any children diagnosed with allergies or asthma. Whereas if you look at other countries, like the United States, that is not true. So let's focus in on the US and look at some data. This is from the United States. And what this shows, this graph, is it shows the rate of infectious diseases over time from the 1950s, and then this graph stops at 2000. So diseases like the measles, hepatitis A, tuberculosis, mumps, all of those diseases went way down in the US over those 50 years, mostly due to increased healthcare and sanitation, as well as vaccination. Okay, so hope you can probably buy that, right? Is that these diseases have decreased a lot in the US. Look what happens if we look at some other things here. This dotted line right here that you see kind of start up from 1980, that's asthma. Look at it skyrocket. Type 1 diabetes, Crohn's disease, multiple sclerosis. As these infectious diseases go down, these other diseases go up. Do you know what all of these have in common? They're immune disorders. They're diseases where the immune system is not functioning properly. So what does this mean? Are these two things related? That's the golden question. So I'm going to show you asthma again over the last 40 years or so. Actually, exactly 40 years. That's cool. And it's showing that if you took the frequency of just the whole US population that was diagnosed with asthma in 1981, it was 3.2%. Over the last 40 years, that's increased to 7.9%, which is huge. We as scientists cannot explain this. It is much too fast of an increase in this population to be caused by chance. Something is happening here. And just FYI, this is the most recent data from the CDC. So it was 7.9% in 2017, 2018, and 2019, about the same. And this comes directly from the CDC. I'm going to skip this slide because it doesn't really add much to our talk. So something's happening in countries like the US. We're seeing a massive increase in immune disorders. So something's happened to our immune systems that's not allowing them to function properly. And that, I think, can be explained by something called the hygiene hypothesis. Yes, we are finally there. This is the hygiene hypothesis. So let me go over what the hygiene hypothesis means. So hygiene, what does that mean? Sanitation, cleanliness, healthcare. I often just use the word dirt, but it means a lot of things. So as healthcare, cleanliness, sanitation increases, which tends to happen as countries become more industrialized and more developed, then the potential to be exposed to microbes decreases, right? Because we just are cleaner and more careful. So we're getting less exposure to microbes in the environment, from other people. We have less human exposure and less animal exposure. So during childhood, if this happens, I hypothesize. So a hypothesis means this is not proven. This is me coming up with a reason of what I think might explain this. So during childhood, the immune system is inexperienced due to this lack of exposure to microbes. Therefore, I hypothesize that the immune system needs to be able to know what a real threat is, like a pathogen, and what an imaginary threat is, like an allergen. And so if the immune system is inexperienced during childhood, then it's more likely to fight an imaginary threat, like an allergy. Have you ever heard that expression like, oh, just let your kid roll around in the dirt, it's good for them. It's okay to eat a little bit of dirt. It's good for them. That is the hygiene hypothesis. If you've ever said that, you have actually talked about the hygiene hypothesis without maybe even knowing it. So if this happens, then this might explain why in places where there's increased hygiene, there is also, at a population level, increased allergies and asthma. I think I'm just gonna, if it's okay, I'll probably just keep rolling and then we'll get questions at the end. All right. So there is some data out there that supports the hygiene hypothesis. It's mostly epidemiology data, which means that it's just observing populations. It's not doing any actual like scientific experiments. So we call that correlation data. It's really good data, but it's not particularly strong evidence. So where my lab comes in, and a few other labs in this country, is we actually want to generate experimental data where we're looking more of causation. Can we more strongly support this idea that this hygiene hypothesis is actually real? So probably the strongest evidence thus far for the hygiene hypothesis, or some of the strongest evidence, is from a study that actually came out of the University of Chicago in 2016. And they did something that was quite genius. So what they did is they actually studied children from two different populations: Amish people in Indiana and Hutterite people in South Dakota. And these Amish and Hutterites have very similar ancestral backgrounds. They have very similar family types. So they have large families with lots of children generally. They have similar health care choices, similar diets, et cetera, et cetera. So they're very similar. What the major difference between the Amish people and the Hutterite people is their farming practices. Amish people use very traditional farming methods. They're using horses. They're involving their children very early on in farming. The Hutterites also farm, but they are industrialized. They use more current farming practices. They also do not involve their children as early on in the farming practices. So Amish, more traditional, kids are getting dirty faster, more, so to speak. Hutterite, more industrialized, their kids are staying cleaner. And what they found in these kids, this is a table taken directly from their paper. What they found is that if you look here at this, children with asthma, they studied 30 Amish kids, 0 kids had asthma. In Hutterites, 6 out of 30 kids had asthma, which is similar to the normal non-Hutterite or Amish population of children. So just the farming practices and exposure of kids early on to farming might be influencing their immune systems significantly. I'm going to, I think, skip the next slide. It's just a little bit more data where they show that it might be due to the dust that is in the Amish and Hutterite households. These scientists actually went to the houses, gathered dust, put the dust up the nose of mice, and then looked to see what happened in asthma. And they saw that the dust from the Amish houses actually reduced asthma levels in the mice, which is pretty amazing. So they did a really great job showing that environment does matter. Let me just go from there. Okay. So I don't want to go over too much. So I'm going to show you just a little bit of what we do in my lab now. And we will skip over some stuff that doesn't matter because it's not my data. And then I'll show you a little bit about what we do. And then I want to make sure we have time for questions. So this work began at the University of Minnesota, which is where I did my postdoc, as Kim mentioned. And we wanted to further investigate how the environment impacts the immune system. And so we used mice to do this. We have mice that are called specific pathogen-free mice, or SPF mice. And these mice are really squeaky clean. They are always in very, very clean environment. They eat food that has actually been pre-cleaned for them. They are just kept really, really clean. And so that's what's depicted in this picture here by this little mouse that's got a little dust Swiffer here. He has very few microbes then because he hasn't been exposed to hardly anything. Now this is in contrast to a mouse that you can buy from a pet store. Pet store mice are like dirty daycare mice. They have lots of microbe exposure. So this is an example of, in this picture here, this mouse that has a lot more going on in his little bubble. So what we wanted to know is what happens if you break this barrier. What if you expose the clean mouse to the dirty mouse and therefore change the microbial experience? What happens to the immune system? So you can study what happens in these mice in terms of their immune responses. This is the model that we use. So how this happens is you buy pet store mice that are dirty and SPF mice in the white that are very, very clean. You can put them in the same cage like this, and just due to being in close contact with each other, this dirty mouse is going to expose these clean mice to microbes. So over time, what you get in this cage is dirty mice that were originally clean. You also keep a cage of mice that stay clean as your control. So you can compare these really clean mice to mice that started out clean, but now are dirty. And I'm gonna skip, because this is not my data. So we do the same thing in my lab now at Grand Valley. And we made sure that our clean mice were clean. So this is what we do. We screen the mice for different types of bacteria and viruses. We want to see if they've been exposed to those things. So you can see our really clean mice don't have any of those things. And then if you see those little plus signs here, those are showing that yes, indeed, our pet store mice have been exposed to those. So you can see we've got clean mice and dirty mice. Even over time, our control mice stay clean, which is important. But our co-housed mice, which are the mice that get a pet store bunk mate in their cages, they get some of these pathogens from the pet store mouse. Not all of them, which is interesting, but at least some of them. So what we can do is we can figure out whether this change means anything for the immune system. I wanted to explain one of the techniques that we use in the lab, but I'm thinking that maybe if someone asks about it, we can talk about it. I'll leave this slide for now and we'll talk about it later maybe. But we use a technique that's called flow cytometry, which is kind of like an advanced CBC machine that can show us the different white blood cells and what they're doing. So it's a really important toolbox for the immune system. So we study in my lab, we study macrophages, which, remember, respond really early on in the immune response. They are like garbage collectors. They go around and they just eat things. So that's how they can tell if there's a threat, and that's also how they try to destroy pathogens. And so we're interested in whether the hygiene hypothesis applies to macrophages. And so what we do is we do flow cytometry, which I know does not mean much right now. Don't worry. What's more important is this graph. What we see is that our co-house, so our dirty mice, the red here is the dirty mice, the black here is the clean mice. We see that our dirty mice have increased amounts of macrophages and that they're more activated, which is interesting. So that means that changing microbial experience is influencing macrophages. So we have continuous studies going on with that particular avenue. And then we also wanted to study T cells too. So T cells are the cells that are like the assassins of the immune system. They're really good killers, and they'll go around killing infected host cells. And so we do flow cytometry there, and we see that we have an increase in the activated T cells in our dirty mice. And these T cells, which are kind of depicted by the green cell here, make a certain molecule called granzyme B that causes the killing of the cells that they're trying to kill. And so the cool thing is you can use this granzyme B to tell if your T cell is a killer or not. And we see that our dirty mice have increased amounts of T cells that are going around and killing. The final thing that I'll mention, because this is what I think our lab is probably most unique for and something that we're really excited about. I wanted to know if microbial exposure, so does the hygiene hypothesis have anything to do with cancer? And at this point, no one has published anything about this. So I wanted to know, like, does what happens to you as a kid in terms of your environment actually influence whether or not you're going to get cancer later? And so we picked melanoma to study in our studies. Melanoma is the most aggressive skin cancer. It's becoming more common. And there's also a mouse model. So here's a picture of a mouse. This is not from my lab, but here's a picture of a mouse. You can actually purposefully give mice cancer by injecting cancer cells into them. So this mouse has a tumor right here. And just like in humans, where melanoma's got a lot of pigment, it can look very dark. These are examples of tumors taken out of a mouse with this mouse version of melanoma, and they're these big black tumors. They're actually very gross. They're very gooey. I don't like them. But we can get a mouse model of melanoma. So what we do is we give our dirty mice tumor cells and our clean mice tumor cells, and then we measure what happens in the tumor. So we can remove the tumors and look at the cells that are in the tumors. And we see we have increased activated T cells in the tumors of the dirty mice. And interestingly, we didn't see much of a difference in the killing ability. So this is something we need to repeat. But we're still pretty excited about this result here because generally speaking, you want T cells in your tumor. T cells are very, very good at killing tumor cells. So if you have more T cells in your tumor, that could mean a better prognosis. Okay, I think I will leave this for now. There's just a little bit more data showing that our T cells in the tumor of the dirty mice are actually dividing more too, like making more of themselves, which is a really good thing. That's what you want to happen. Okay, I know that was a lot. I was wanting to share with you some of the stuff we actually do in my lab, so you can see what's happening, right down the road at Grand Valley. So in terms of the conclusions, kind of what are the takeaways from this talk? I think that the hygiene hypothesis is real. I do think that our data that's unpublished, as well as some other people's data, shows that the environment really does impact the immune system. And we in my lab have seen that to be true for macrophages and T cells. So who knows what else is also impacted by this. We also think, and this is what's exciting to us, is that this may have an impact on tumors, which has not been explored at all yet. So we're going to follow up with a lot of studies with that too. And so altogether, we do in my lab have evidence supporting the hygiene hypothesis that you want to be a little dirty so that your immune system actually develops properly. So any questions now? So this, I'd be happy to take any questions. I can go back, to spots in the PowerPoint. I'll kind of let you moderate, Kim. Buckley.
Great, Kristen. Fascinating work in your lab. Holy cow. Yes, we have several questions stacking up in the chat room, and I'll just take them from oldest to youngest here a minute. First one is from Greg Sewell. Does the second pathogen exposure increase the immune memory level? Which I think you already answered. And then a follow-up to that was, can T and B cells have memory for more than one pathogen?
A single T cell cannot have memory for more than one thing. It is specific to one thing. But you've got more than one T cell, and they're all different. So as a population, yes, you can have lots of different memories. So that's like when during childhood vaccinations where kids are getting, vaccinated with different things all at once, that's totally fine because you can generate the memory, T and B cells to all those things at once.
Great, thank you. Then from Nan Carr, she says, I started with seasonal allergies in my 50s, causing seasonal asthma. Why then? And then second part of that is my husband, who suffered from hay fever much of his life. The symptoms stopped in his 50s. What is happening to cause this?
Immune systems with age, which I think is what my next lecture is, isn't it, Kim?
Yeah, something like that. Yeah.
Yeah, so that's due to changes with the immune system with age. So a lot of things happen as we age with immunity. And without going down a huge rabbit hole, there are certain things that break down with age and allow immune system to respond to things it's not supposed to. So that might be happening in your situation. And then there is also evidence that shows that certain immune functions decreased with age. So I could see it either way, depending on the individual, that it makes sense to me that, you might have increased allergies or even pre-existing allergies that then decrease, just depending on what happens to the immune system as it ages.
Great. This question is from Ben Hancock. He says he's read a great book on this subject called The Microbiome Solution by Robin Chutkin. And she makes some of your same points. Do you have any published writings or other reference works to suggest?
I don't. I wish I did. I can think about that. Let me think about that. I might be able to send some links along that then, Kim, you could pass on. Most of the stuff I have are like primary literature reviews.
Right.
But I think there's a few good ones that that might be interesting for people to follow up with.
Okay, great, thank you. And I'll be happy to pass those along.
Yeah, I'll write a note to myself.
Here's another comment. You may have already answered this. It says, I got asthma in my teens. I stopped having triggers in my 40s. Do I still have asthma?
It depends, honestly. So there are cases of severe chronic asthma where exposure to allergies are no longer needed. Like you can be taken away from the situation or the environment or whatever that it makes you have asthma, and there's so much inflammation and damage done that it just keeps going. And so it would kind of depend on the severity of your asthma early on, and what the allergen was, so the answer is maybe, but if you don't experience symptoms, like even if it's induced by like exercise or something, then you might not have had chronic asthma. There is asthma and then there's chronic asthma. So.
Good deal. Yeah. Next question from Ben Hancock for you, Kristen, is how are you able to engage your students in your lab?
Oh, that's a good question. So how did I before COVID, maybe is a better question. So I like having a good number of students involved. So for instance, usually I have about 2 master's graduate students who are working on their theses. And then I usually have anywhere from 5 to 7 undergrads that come into the lab with me. And we work as a team. And so we develop together the questions that we want to answer. And then I help them with the scientific method to figure out what's the actual hypothesis. How do we design a good, well-controlled experiment? And then when it comes time to actually do the experiment, I train them how to do the technique. And what my goal is usually for students, particularly my master's level students, is I want them to get to the point of feeling more independent, where they feel confident in thinking about the science and doing the science. And so I'm not a micromanager. So yeah, so I'm heavily involved, especially from the planning and the training. And then, I'm happy when a student comes to me and says, hey, look, I did this experiment and have this data, and we get to think about what that means together. So I kind of engage with them on every level, but I do like to push them towards independence.
Great. Next question from Greg Sewell. Would you recommend the book entitled Let Them Eat Dirt?
I haven't read that whole book, so I couldn't make a good recommendation for that. I would say that I am in no means wanting to stop like vaccinations or our current healthcare practices. I mean, we have a very good reason to do the things that we do in this country, especially. And sometimes I feel like some of those books go kind of to the extreme, but yeah, I can't, I guess my answer is I don't know. I can't recommend that one either way.
Okay, the next one from Bill Bredemeyer. So are you saying it's healthier for everyone to be a little less worried about dirt, hand washing, the 5-second rule about eating food dropped on the floor?
To an extent, right? Yeah. Yeah, this is hard. This is a tough one, because I mean, obviously hand washing is very important. But yeah, like with my kids, we don't really do the 5-second rule unless it like literally drops into poop or something. But yeah, I don't know. It's hard to take what we find in experiments at this stage and then make like practical, useful suggestions for human health. That's the hard thing, is that I want to be able to say things like, if you do this with your kids or your grandkids, they're going to be healthier. And we just don't have enough data to suggest what those suggestions would be. So, but yeah, I think that there's a good, healthy exposure to other people if you can. I mean, in these times it's hard, but if healthy exposure to others, to new environments, and not bleaching your child. I've actually seen some parents like bleaching kids' hands immediately after they touch something, and I don't know about that.
Thank you. From Don, how do the Amish and other farm communities compare in terms of rates of childhood immunizations?
Yeah, good question. So the Amish and the Hutterites both have equal and very, very high rates of vaccination for their children.
Excellent. And from Myra Karachi, not sure I'm pronouncing that correctly. Are you worried about today's kids and hand sanitizer, etc.?
Yeah, I am. But all of there's, with any of these decisions regarding health, there's going to be obviously potential detriment, right? And I am interested from just a purely scientific standpoint to see, like, if we were to look at toddlers and babies that are around right now during this past year, and then like in 10 years, see what their asthma rates are, because I would argue that everybody is being cleaner. I mean, I know my own family, no one has, with our mask wearing and social distancing practices, like no one in my family, including my kids, no one's even gotten a cold over the last year. So we're preventing ourselves from being exposed, right? And so it's going to be really interesting to see, what the rates are of like asthma and stuff with this population of kids. So yes, I am, I don't know if I'm worried, but I'm interested really to see what happens. And with the hand sanitizer, like, we know that hand sanitizer is very good at killing, it's actually inactivating the SARS-CoV-2 virus. So since that's been our primary focus, I think the hand sanitizer has been necessary for that. But if it were not COVID times and we were using hand sanitizer like we are now, I would be worried. I don't know if that makes sense, but it kind of depends on what the priority is.
Good, thank you. From Kearney, can you expand on the dust exposure of the Amish versus the Hutterites? Seems like the dust itself could be an explanation of differences in the immune responses.
Yeah. So something, yeah. The short story, the short story of that is we don't know what's different about the dust. Some people argue that being exposed to lots of different types of dander, you know, and potential allergy causing things. Some people argue that if you get that really early on, that your immune system will better know that's not a threat. I don't need to respond to that, if that makes sense. Like you're exposing yourself to stuff early so your immune system is trained properly. And so because in the Amish, there are increased exposure to like farm animals, that may be why that's something in the dust, you know, that gets carried. But we actually don't know. I think that lab has actually taken the dust and like done a lot of analysis to see if they could figure out what the major differences were, but I'm not sure if they've even published that yet. Yeah, so yes, something is in the dust, could be doing something, but we don't know what or what yet.
Thank you. From Marilyn Schmidt, do you concur with the suggestion that antibacterial hand soap is not recommended?
That's a dicey topic. Yeah. So antibacterial hand soap is called antibacteria. This is probably more information than what you want to know, because it had triclosan in it. And triclosan is a chemical that had some antibacterial activity. Because it was being used so much, we actually started getting populations of bacteria that were resistant to triclosan, which was a problem. So because of that, the FDA wanted antibacterial hand soap to be taken off the market. And so what happened, and I think that has trickled down most of it now. You don't, if you have, I mean, there's not a lot of soap that says antibacterial on it anymore. And so that's why that happened. And I agree with that. Like, and I don't think you can get antibacterial hand soap. And that's because of more of influencing the bacteria itself. Soap, what it does is it washes away through mechanical action. And I think that hand washing after, you know, I think hand washing in and of itself is a good thing.
Great, thank you. From Lance Everett, when my husband goes outside when it is humid, he gets a tight chest and trouble breathing. The doctor said he doesn't have asthma. Is it an allergic reaction?
I don't know. Please remember I'm a PhD, not an MD. Yeah, I feel uncomfortable speculating.
Okay, and the next one from Kearney again. Can you imagine a future where we have vaccines against MS, diabetes, or Crohn's?
Yes. Yep. They're working on them.
Now that's encouraging. There is work being done on them. Excellent.
Yeah, there's an MS vaccine, I think in phase 2. Yep.
Excellent. From Nankar, are there any studies that show there are supplements that can boost the immune system, such as echinacea?
The data, scientific data does not support that supplements impact the immune system. So that's all I'll say.
Okay.
I think that's a whole nother topic.
All right, and this one final comment, I think, from Alda is to a previous question about the light, tight chest and trouble breathing. Comment is there is less oxygen in humid air. Okay, so that does it for the chat room at the moment. We can open it up to any other questions.
I can stop sharing this too for now.
So this is a question from Greg. What is the best way to have a strong immune system?
Sleep, eat healthy if you can. Be vaccinated because that helps, boost your immune memory. Regular healthcare, kind of the things that just you do to be a healthy person anyway. I can't really, based off of scientific evidence in peer-reviewed journals, I can't recommend any like supplements or anything. The data just does not support that if you take vitamin C or something that you're gonna live forever because your immune system is perfect. That's just not true. There's not there, so it's hard to find a miracle drug. I think the thing that we see, as people get older, and I'll talk about this a little bit in my next lecture, but we see this danger of frailty where people become frail and they aren't able to move around as much, kind of get stuck. And that's actually happening a lot in right now times too. And that is like a switch for the immune system to tank if that happens. So, really trying to stay healthy and active as much as possible is really important too, just to prevent frailty from happening.
Great, thank you. Kristen, I have a question related to vaccinations, COVID and the vaccines specifically. As people, as a wider swath of the population becomes vaccinated, what's your thinking, if any, on the ability of those fully vaccinated people to transmit the virus to others? Any thinking on that?
Yeah, that's actually, not even thinking. That's been really well shown in studies, both with this vaccine and virus, as well as other related vaccines and viruses. So if you get infected with the virus, if you're not vaccinated, that virus is allowed to replicate. We had that on the graph. Replicate and replicate and replicate till finally the immune response comes in, hopefully, and curbs it. In a vaccinated person, the virus gets in and starts to replicate. But that replication is curbed. It is halted much faster because of the memory immune response. If you have less virus around in your body, you are less likely to transmit it to other people. And that's been shown time and time and time again. It's exactly why we wear masks too. So we know people with higher viral titers, therefore much higher viral replication. We know that they are the major transmitters of the virus. So if we reduce the number of people who become major transmitters because of vaccination, transmission rates are also going to go down.
Great. Thank you.
Mm-hmm. Yeah, no problem.
Another question in chat from Sue. Does any of this relate to Parkinson's?
I don't know. As in like an immune response in Parkinson's, or I don't know what that means. I guess let me just say I don't know enough about Parkinson's to talk about like the immune response that's happening there or what it might mean. I'd have to look into it. Yeah, I'll leave that there. I know more about MS because I use that as an example in teaching, but I don't know as much about Parkinson's.
Okay, good. And then from Bill Wells, I'm not sure if I understand this question. It's simply stated as relate to autoimmune diseases. So I don't know if there's anything you care to comment on.
Yeah, so we know that autoimmune diseases are also increasing in countries like this. Actually, in that graph I showed, the second part of it, there were some autoimmune diseases represented on there, like type 1 diabetes, for instance, that are going up. So I'm really interested in understanding, whether training your immune system early on is also going to prevent potentially autoimmunity, because autoimmunity is also where the immune system is not functioning properly. It's responding to an imaginary threat. It's not supposed to respond and start killing off your own host cells. Like, that is something the immune system is absolutely not supposed to do. So maybe proper training earlier on is important for preventing that from happening. And there are mouse models of autoimmunity too, just not something we do.
Excellent, thank you. Another question from Sharon Arnshorst. What is your opinion about the added levels of air filtration systems that are now being added as extra systems to furnaces? They are promoted to cut out bacteria in the home air.
I don't know. Again, it's one of those things where, like, we just don't know enough yet to influence actual action. So, because we're still, that's the cool part of this research, is that, like, you're seeing, like, early on the cusp findings, but we don't know enough yet. So yeah, I don't know. I don't know why you would have, like, a ton of bacteria floating around near your furnace anyway. Like, that seems, you know what I mean? Like, that maybe seems unnecessary, but for all I know, we probably have one of those in our house too. So I don't know. Sorry.
Okay, that appears to have answered all the questions in the chat room. Anybody else? Comments or questions?
I have one. If you look at the amount of resources that have gone into addressing the COVID, it stands to reason if we were able to marshal similar resources, we could address through research and ultimately the development of vaccines so many of these other diseases that we face. Do you feel that there's going to be any momentum coming out of the post-COVID era to continue some of this in terms of funding for NIH and other places that might speed up the process and the timeline for other vaccines?
I hope so. I really hope so. Yeah, it's tough because for some of the vaccines that we've been struggling with, such as an anti-malaria vaccine and an anti-tuberculosis vaccine and an anti-HIV vaccine, for instance. Those are probably the three that pop in my head that people have been working on vaccines for years and years, and it's just we're having a hard time. There are other reasons why those are so difficult to establish vaccines too. So having this really cool technology that's actually been developed, it was developed over 10 years ago, this mRNA vaccine technology, but having this now in use regularly, I'm not sure if that's going to influence some of the vaccines that we as a scientific community have struggled with. But if we have another outbreak of some type of virus that's similar to what we've seen with SARS-CoV-2, I think that we will be able to much more quickly produce something and get it under control. So, and I do hope, like you said, with the funding situation, that's always what it's about, right? I'm completely funded by grants. And if I don't get grants, my research stops. my students can't work, whatever. And so if I can't convince someone out there, usually it's a committee, that my research is worth doing, then I don't do it. So I hope that this does renew continuous research into immunology and into vaccines, just in general. So we'll see. But yeah, I hope so.
Well, and I think it may also encourage young people to want to go into the field with as much visibility as the whole field has been given.
Good point. Yeah.
Well, Kristen, this seems like a good point to maybe wrap it up. I think you've done a great job here. Good for you. Well done.
Oh, thank you. Thanks for having me back.
You betcha. Well, we're going to try to get you back here a third time at some point in the next year or two. So
Thanks so much, Kristen.
Welcome.
Have a good day, everyone.
thank you.
Thank you. Good job.